All right. Give me a thumbs up if you can see my slides. Awesome. Okay. So this is our Spring Observer Refresher. Thank you for joining. I'm making the assumption, and I know most of you, this is a safe assumption, that you have either had a prior training or you have observed in the past. So we're not going to go over a lot of the basics that we've covered. So we're just going to jump in. We are going to review a few things. The Nature's Notebook website has been updated and has a fresh look. I think that happened late January. So I'm going to walk you through where to find things on the website. And then we're going to spend a lot of time talking about the phenophases, make sure we're all up to speed and good on those. And then there will be plenty of time for you guys to have any questions answered. So just because I know some of you haven't had a formal introduction to our organization, I am Amanda Wanless. I am the Executive Director of Indiana Phenology, and our organization really is focused on exclusively on engaging volunteers of all ages in documenting the timing of phenology in Indiana. And we really want to understand how our environment is changing and how species are reacting to these changes. So what you guys are helping us do is collect that baseline information and that change over time information so that we can really explore these types of questions and have a better understanding of our ecosystems here in Indiana and what's happening with them. In case you weren't aware, we, in addition to facilitating our citizen science phenology observations, we do environmental education programming and we collaborate with community partners. And we have 3 programs. I know some of you are Indiana Backyard Observers. Some of you observe at some of our phenology trail sites. We also have a schoolyard program where we help teachers engage their students in making observations on their school grounds. So talking about phenology today, as you all are aware, phenology is really those seasonal changes. It is the recurring plant and animal life cycle stages and also the study of their timing and relationships. It's happening all the time, happens everywhere that they're living creatures, and it really can be observed on many different scales. And just to give you the boil down, here are why we study these seasonal changes, why we care about these. The environmental conditions that a plant or animal is experiencing at any given moment in time, it's very local conditions are what help it know what it should be doing. We look at the clock, we look at the calendar to decide what we should be doing at a particular point in time, but the plants and animals, they look at the weather, they look at the conditions surrounding them to help them know what's happening. And we are seeing changes in our climate, in our environment, and those changes are changing the timing of the phenology. And when the phenology timing changes, that has 2 different types of impacts. It changes the interrelated components, the relationships in ecosystems, and then it also affects the humans that depend on those ecosystems. So that's kind of the background of why we're making these observations. And to reiterate, we are seeing changes in our climate. So I thought as it is spring, and that we're going to focus on what we're going to be seeing this spring, that we could start by taking a look at how spring is coming along in the US and in Indiana. So I've got some fun maps here. These are from the USA National Phenology Network. These are their status of spring maps, and they put these out each year. And it is a map that is based on a model that looks at the weather and temperature and the trends. And they put a new version of this out daily. And it predicts really when spring is going to start or when different happenings are going to start in various different locations. And it's based on Nature's Notebook data on looking at honeysuckles and lilacs, cloned honeysuckles and lilacs, so that any variation in the timing is not due to different genetics, but is due to different environmental conditions. And they've got a data set of this that goes back at least 50 years. And so they use that to put out these maps. And what this is it's the spring leaf index anomaly. And I pulled this a couple of days ago on March 6th. So it has changed and spring has spread a little bit farther than when I grabbed this, but we've got two different scales here. It shows us how, oh, let me put the right words to this. Okay, so it's got two different scales. It shows us how early or how late spring has arrived to a particular location compared to a 30-year average. So the red is looking at late, or no, excuse me, the red is looking at early, and the blue scale is looking at late, and the darker the color, the later or earlier it happens to be. So you can see a little bit of blue on this down in Texas and Florida and out west in scattered patches, but a lot of red, which indicates that there are some places where spring is a little bit late compared to the normal, but a lot of other areas it's early compared to normal. And you can see up in Indiana, we've got some dark red, which means that spring is 10, 15, 20 days early compared to that average. So early spring here in Indiana is the takeaway. This next map, this one, instead of looking at leaf out, which is what the first one was looking at, this one looks at bloom time. And again, I haven't seen any lilacs or honeysuckle blooming yet here in Indiana, and that matches with this map that it shows that the bloom hasn't spread that far north yet. But again, what we see a little bit late in spots of Texas and Florida, earlier in a lot of the other areas where spring has reached. But the question when you see maps that should immediately pop into your mind is, how unusual is this amount of variation? Because spring goes back and forth. Sometimes it's a little earlier, sometimes it's a little bit later. So this next map answers that question. So this compares the leaf out anomaly map that we were looking at just before to show how often we see that amount of variation from the average. And we've got, again, two color scales. The blue relates to the late, and the green relates to the areas that were early. And the places where you see gray, that's where it was dead on average. And so you can see there are a lot of places in the country where spring has arrived, the spring leaf out, that are pretty normal. And then in a lot of the other areas that were early, what you see is that it's not that unusual. The lightest green on here is we see earliness that often, 1 to 4 years. So that was tongue twisted up there. The light green, we see that amount of earliness, that earliness every 1 to 4 years. So it's fairly normal amount of variation that we're seeing across most of those areas here. The darker greens over in Missouri and Illinois, some of that is a lot more unusual, but here in Indiana, Kentucky, on down, it's pretty darn normal. And if you look at the spring bloom that hasn't yet reached us, again, there you're seeing that this is pretty normal spring. Even though it's early, it's a normal amount of variation. So that's how spring's shaping up. Let's jump on into looking at making observations and reviewing how we do that. So general process, go outside, look around, share what you see. That's pretty familiar to you. We use 2 different platforms. I know some of you have been around for a while. We focus on Nature's Notebook. In the last year or so, we've added iNaturalist and are really starting to encourage our observers to use iNaturalist to supplement Nature's Notebook observations. So if you're not familiar, iNaturalist, some people like to think of it as an online social network for people sharing biodiversity information. So you can share information, observations that you've made, and then you can talk to each other and engage with others and learn from each other. We like to use iNaturalist as a way to capture photographic evidence of phenology. So Nature's Notebook, you can't add those pictures, but if you use iNaturalist with it, you can take pictures of those phenophases, what's happening. And then you've got those two different ways of using it. Plus, you can also make observations of the phenology of things that aren't in your Nature's Notebook observation site at the same time. So if you haven't ever used iNaturalist, check it out, and I think you'll like it, and it'll complement Nature's Notebook. Here we are, Nature's Notebook. It is for looking at change over time. So we're going back to those same plants and animals. We've got our data sheet, those phenophase questions, which are really the core of what you do, making observations. And you can either submit your observations through the app that's in the middle, or you can submit your observations through the web interface. Give me a little bit of feedback here. How many of you use the app?
I do.
Okay. Does anyone use the web interface to submit their observations? Okay. My recommendation is using the app. It is the easiest, most friendly, and shortest number of steps. But I do want to give you a brief overview of the web interface, just so you know what's available there, because there are some things that you can see on the website that you can't get to in the app. All right, so this is what their new website looks like. Your observation deck is, the way you get to it is right there when that little arrow spot banner that says your observation deck. So you click on that, and then it takes you to this page. So previously, your observation deck looked a lot like this, but it was all kind of spread out down the page, and so you'd have to scroll down to the bottom to see this part. They have just reorganized things and organized them into different tabs. So now you've got an observation tab, which is where you did most of your stuff previously. And then they also have a learning tab that has some resources that might be useful for you. They have an observer certification course, I think they call it, and some other things that you can get to. So that's something you might want to check out. Also, now from your observation deck, you can get to the visualization tools. That's that third tab there. So if you click on that, you can go and you can make phenophase calendars with your data, and you can interact with any of the data that anyone else has submitted. And then there are some other tabs there. So just really quickly walking through how this works. Up at the top, you've got My Account. If you need to change your settings, your password or something, you can go to My Account to do that. Then it has on the main observation deck these 4 panels, and the choices you make on the left dictate what shows up in the other panels. So you've got your first panel. If you are a backyard observer and so you created your own site, you would want to select the spot that says personal sites. If you have your own sites, but you also are a trail observer, then those will all be listed there. And depending on which site you pick, which group you pick, that'll change what shows up here. So I've selected my personal sites, and I have a backyard area, and then I have a front yard area that you're seeing there as well. The second one shows all the plants and animals that I am observing at the particular site that is selected. So at my backyard site, there are some options down there. This is where you would go if you want to change the order of your plants and animals. So if you want it set up so that on the app, it shows you the first plant that you come to, and then the next plant, and then the next plant, you can use the sort option here to change what that order is. So that's really handy to know how to find it. You can only do that particular thing on the website. You can't do the sorting in the app. And you can also print field data sheets through this particular section here if you want. This is what the field data sheets look like. since none of you use that, I'm going to jump over that and not go into it more. The third panel gives details for whatever organism you have selected in the plants and animals tab. So right here it's showing Forsythia. And two things that are useful to note here, it has an option for viewing the species profile, which gives you all of the phenophase definitions and optional questions for whatever organism you want. So if you wanted to have that printed out so that you had it to refer to while you were out observing, you could print that right there. They also have an option to download photo guides for the species that they have photo guides created. Nature's Notebook doesn't have a photo guide for all of them. We do have on our website phenophase photo guides for a lot of other species. So Nature's Notebook ones you can get to here. I'll show you a little bit later how to get to ours, but those can be helpful because it shows a picture of each of the phenophases. And then the last section here is entering observations. Okay, so this is what it looks like when you're entering observations. I'm going to just skip over those since you guys don't do it that way, and the app is just so much easier. Okay, so quick review of the app. They have made some updates to the app recently. They do that periodically. Sometimes after they make updates, you have to go back in and re-log in and then make sure you grant access to Nature's Notebook app to access your account. That's there. Again, the app is organized a lot like the observation deck. It's got up at the top right here. There's the navigation. So it's up at the top. It's got this navigation menu. The choices that you make on the first tab change what options you'll see on all the other tabs. So on the first tab, that's where you select which group you want to be observing with. In this case, I'm picking my sites or my personal sites, and then that would show what personal sites you'd have there. If you're a backyard observer, if you're a trail site observer, you'd make sure you pick whichever one it is there. What options show up here are specific to what groups or sites you have joined or signed up for. And then on the plants and animal tab, that just shows you all the plants and animals. Down here, there is this button where you want to, if you want to add plants or animals. If you have been observing for a while at your own site and you just keep thinking, oh, there's this other plant I want to observe or this other species, you can add that either through the app or the website and then have that additional species as something that you make observations of. And then the main tab of interest here is this third tab, the Observe tab, and this is essentially the data sheet. You can see up there are 3 different buttons or tabs along the top that say plants, animals, and site visit details. So if you only are observing plants, then you'll just be in this plants tab answering these questions. When you click next, that takes you to the next plant. You enter all of your answers, that takes you to the next plant, and you enter your observations and so forth. If you have animals, you'll want to click over to the animals tab and make your observations there. And then the third one is for site visit details. And those are answering questions like how long did it take you to observe? Was there snow on the ground or snow in the trees? And those are observations or information that will apply to every one of the other observations that you've made of the plants and animals. And then I believe all of you are familiar with this lovely little eye in a circle button, but this is super helpful because it gives you that phenophase definition for that question, that phenophase question. And then it gives you those optional questions that you can answer. And I've been observing for 6 years now. I still click that little button to make sure that I am answering the questions correctly, because the more closely we answer a question to that definition, the more consistent our data is, the better quality our data is from observer to observer. All right, so let's just get past these right here. Let's review the process of making observations and jump into the phenophases. So again, no surprises here. When we're making phenology observations, we want to try to go out once a week at least. We want to visit the selected plants and animals that we have at our site and then submit that data. We're going to use this data sheet, essentially just answering these yes or no questions about the leafing, flowering, and fruiting of the plants or the activity of the animals that we have selected to observe. And you answer yes, no, or unsure. The unsure is a perfectly valid option, and there will be times when that is the right answer to pick, and that is totally okay. So don't be afraid of using that when you need to. So let's look a little bit more closely at the phenophases. And there are two different sets of phenophases. There are those that have to do with the leaves, and then there are those that have to do with the reproduction of the plant, the flowers and the fruits. And so we'll look at both sets. And I am going to go through and I'm going to read these definitions to you because I think it's really important that we go back to those definitions. So let's start with the first one, breaking leaf buds. And the definition is one or more breaking leaf buds are visible on the plant. A leaf bud is considered breaking once that green leaf tip is visible at the end of the bud, but before the first leaf from the bud has unfolded to expose the leaf stalk or leaf base. And so we have three pictures here. Only the middle picture here shows breaking leaf buds. The first picture on the left shows our dormant buds. And if you go to the definition, it has in it some words that you can take as like a cue for when you start observing this and some other phrases that are your cue for when to stop. So our cue for starting in breaking leaf buds is when that green leaf tip is visible at the end of the bud. So in the dormant buds, the leaf scales are tightly closed and overlapping, and you see no green leaf tip. Right now, there are several different types of plants that have are in the breaking leaf bud stage, and so they have those leaves starting to peep out. But there are a lot more plants that are still dormant or still really tightly closed. So this is the one that we're going to start seeing a whole lot more of through the rest of March, April, and even into May. You'll be seeing breaking leaf buds on most species. And then we stop saying yes to the breaking leaf buds when we see that that leaf stalk, that stem and the base attached. So the last picture here, that leaf has sort of unfolded and we can see the stem where it hits and the twig. And so that last picture is a no for breaking leaf buds. And a hint here, there are some species that the brand new leaves kind of have a reddish tint. And once you see that fresh leaf tip peeking out, even if it's sort of reddish, you're still going to answer yes to breaking leaf buds. Don't get hung up on the green for those particular species where its new leaf is a little red tint. All right, the second one, the leaves phenophase. This is the one that you're going to say yes to for the longest period of time. This essentially captures the growing season on that particular plant. The leaves are that structure that really helps the plant make its food and undergoes photosynthesis. And so the leaves phenophase is trying to capture the entire period of time that there are live functioning leaves on the plant. And all four of these pictures here are all yeses to leaves. So once that leaf has unfolded, it's out of its bud, all the way until it falls off the plant or dies, you say yes to the leaves phenophase. And I don't think I read it yet, but we'll go ahead and read it. One or more live unfolded leaves are visible on the plant. A leaf is considered unfolded once in its entire length has emerged from a breaking bud, stem node, or growing stem tip, so that the leaf stalk or leaf base is visible at its point of attachment to the stem. Do not include fully dried or dead leaves. So if you have a branch that breaks and there are some dead leaves on that, you don't count those as leaves anymore. I think the useful thing to note here is it's that entire leaf and that entire stem has emerged from its bud. It's all visible. And so leaves, they're going to be on there for the longest period of time. Increasing leaf size is kind of a subset, a period of time in the life of the leaf. And this one requires a little bit of context or background knowledge. So the definition is a majority of the leaves on the plant have not yet reached their full size and are still growing larger. Do not include new leaves that continue to emerge at the ends of elongating stems throughout the growing season. So for this one, you have to know what full size looks like, and you have to be able to make a judgment of whether the majority of the leaves, half of them, more than half of the leaves are full sized yet or not. Once most of the leaves, half of the leaves are the majority, so more than half of the leaves are full sized, then you start saying no to this. So this is one that is most typically going to be exclusively a spring phenophase that you'll only see in the spring. There are some circumstances where you might report increasing leaf size later on in the season. For instance, if something happened to the plant and it lost a whole bunch of its leaves, and then it started a new period of growth and leaf out, which may happen if a plant was diseased or it had some sort of insect attack where it was largely defoliated. Sometimes when you prune or otherwise cut branches off a tree, that will provoke new growth. And in those cases, there might be a time in which you would start reporting increasing leaf size again. But again, it's most of the leaves, more than half of the leaves are full-sized, or are they not? And that's how you decide whether you report yes or no to this one. The next one is colored leaves. One or more leaves show some of their typical late season color or yellow or brown due to drought or other stresses. Do not include small spots of color due to minor leaf damage or dieback on branches that have broken, and do not include fully dried or dead leaves that remain on the plant. So this one, and all of the other ones as well, one or more is enough. One is enough for you to say yes to this. So if you see one leaf that is its typical fall color, or you see one leaf that is starting to yellow or brown, that's enough for you to say yes to colored leaves. So we typically think of colored leaves, seeing those in the fall, but you will see colored leaves, particularly on specific species or on stressed plants at other times in the year. So look for this even when it's not fall and you're not expecting to see it. I don't think there's anything else I need to say about that one. All right, so that brings us to the end of the leaf phenophases. Now, in Nature's Notebook, they divide the plants up into kind of two different categories based on whether it's a woody plant or it's a non-woody plant that dies back down to the ground, or an annual. And so we talked about the breaking leaf buds phenophase for those deciduous woody plants. But for things that are perennial that die back down to the ground, herbaceous, not woody, the corresponding phenophase is initial growth. And this is, it's very similar. It is, again, looking at when that green leaf tip, that green growth pops up, and it ends at the point at which those leaves are fully unfolded as well. So those two are very similar, but the particular definition that you are using is going to be, if you click that little I button, it'll tell you the definition, and you focus on that particular one. But it has the same sort of start and stop cues. All right, so now let's jump into the flowering and fruiting phenophases. So 3 of these are related to the flowering, and then the other 3 relate to the fruiting, and this really is the reproductive stages of the plant. And so the first one is the flowers or flower buds phenophase. And like the leaves phenophase, this is capturing the entire period of flowering on the plant. So for this one or more fresh, open, or unopened flowers or flower buds are visible on the plant. Include flower buds or inflorescences that are swelling or expanding, but do not include those are tightly closed or not actively growing or dormant. Also do not include wilted or dried flowers. So once those flowers are wilted or dried, that's the end point. And then the beginning point is when those flower buds are swelling, they're active, you can tell that they're flower buds. Now, some plants that flower early in the spring, some trees that flower earlier in the spring, their buds are dormant. You can see those buds on the tree even in the fall after all the leaves fall off. The dormant buds are not ones that you say yes to. It's once it starts to swell and expand and it kind of changes its color. It's not necessarily totally pink, but you can tell a distinct difference in the quality of an active flower bud, yes, stage versus those dormant buds. Okay, next one. So open flowers, this phenophase is a subset of the flowers or flower buds stage, and this is looking specifically at the open flowers. So this is the point at which those flowers are active, available for pollination. So 1 or more open fresh flowers are visible on the plant. Flowers are considered open when the reproductive parts are visible between or within unfolded or open flower parts. Where those are located is going to vary depending on the particular form of the flower for the species you're looking at. And you don't include the wilted or dried flowers. And then the last 2 phenophases right here that I want to talk about are the fruits and the ripe fruits. And we're going to look at these together. Now, these are so variable what the form of the fruit takes on various different species. The way the fruit looks is how you would tell species apart, one big way of telling them apart. And because they are so different from species to species, in Nature's Notebook, in the definition, it has specific species, specific information. So I've got the definitions for tulip tree here, and these definitions will be a little bit different depending on whatever species you happen to be looking at. So this is a good one to make sure you double check that definition on. So for tulip tree, 1 or more fruits are visible on the plant for the fruits phenophase. For tulip tree, the fruit is a winged seed tightly grouped with many others in a cone shape that changes from green to tan or brown. Do not include empty cones that have already dropped all of their fruits. So this picture right here shows an unripe fruit, and then it also shows the ripe fruit. And anything in this range is something you're going to say yes to for fruits for a tulip tree, because that's what those fruits look like. The ripe fruits is 1 or more fruits are visible on the plant. For a tulip tree, a fruit is considered ripe when it has turned tan or brown, and you don't include the empty cones that have already dropped all of their fruits. So some things that are consistent from species to species, that it's 1 or more fruits are visible on the plant, or 1 or more ripe fruits are visible on the plant. And then when it's empty of seeds or fruits, you stop reporting a yes to it. And so these 2, again, overlap. The fruits shows you the entire period of time there is fruit on the tree, whether it's developing, fresh, newly developing fruit, all the way into its ripe fruit. The ripe fruit phenophase is just a subset of that. There's 2 other phenophases I didn't specifically detail here. One is pollen release. That is a phenophase that is only observed for species that are allergens. And basically it's you can see pollen, visible pollen being released. And then the other one is recent fruit or seed drop. And that is that fruit has been removed or dropped from the tree since your last visit. All right, some notes here about phenophases. They overlap. I've said that a couple of times now. They'll overlap, they'll occur at the same time on a single plant. So right here is kind of just a picture of how the leaves may overlap for a given species. The length of the bars will vary depending on which species it is, but breaking leaf buds is first. Typically that'll be at the beginning. Sometimes you'll have other periods of breaking leaf buds scattered around, but you'll most often see it at the beginning of the season. Then you've got your increasing leaf size, and then you've got your leaves, and then colored leaves and falling leaves will be down at the other end. They'll overlap. You answer each question independently. You can say yes to multiple phenophases at the same time. This is normal. Similarly, the fruits and flowers phenophases will overlap. You'll have flower buds and open flowers at the same time. On occasion, you'll have ripe fruits and unripe fruits, recent fruit or seed drop. All of these things can overlap, and that is totally okay and normal. I want to point out here with this slide that different species show different patterns in the timing of the phenophases. So right here, we've got two very different plants. We've got a spring ephemeral, Virginia bluebells, and then we've got common milkweed on the right. And these are phenology calendars. These were printed using the visualization tools in Nature's Notebook that you can get to through the observation deck. And on the bottom, it's just a timeline, a calendar, January through December for both of these. And I have just plopped on there a bar across June, just so you can see the differences in these. And spring ephemerals, they take advantage of the open canopy, the trees not having leaves on them yet, to pop out, flower, and set seeds and complete their life cycle before the canopy fills out. And so by June, they're pretty much done. Whereas common milkweed has a very different life cycle strategy, life history, and it's just barely getting going when we are starting June. And so the order of these phenophases that they occur in for one species is going to be very species specific. It's going to be a different pattern, a different order for other species. So for both of these ones here, you get the leaves first, then the flowers, and then the fruits. Sometimes you will see species having their flowers first and then their leaves second, and then their fruits come later in the season. Some that are like that, oaks often have their flowers out before their leaves or about the same time as they're getting leaves. And redbud is another one where you're going to see those flowers, those bright pink flowers, before you get the leaves. And so the particular order that these things happen in and the overlaps that you might see is going to be different depending on what you're looking at. Just something to keep in mind. All right, so phenophases. The phenophases really are the key to understanding what you're seeing. And some things to pay attention to: if you can recognize the leaves and the flowers and the fruits of your plant, then you are good for making observations. You really need to be able to recognize those different structures and how they look, be able to distinguish between them, and then understand the order that you expect to see them to really make good observations. So here's redbud, like I said, there's its flowers, there's its fruits and its leaves. Once you know what each of these different structures looks like, that really makes things clear when you're making observations. You know what you're answering your questions to, but it also helps you to be able to identify eastern redbud when you may see it someplace else. There's really the key to understanding different plants. And so I want to take you through two different, we'll call them case studies here, two different species, and look at how understanding what the leaves and the flowers and the fruits looks like can help us answer those phenophase questions. So celandine poppy is a spring flower, one that we're going to see coming up soon. And this picture on the left, this is a very early picture of it. Its leaves are still kind of folded up. They're still unfolding. And then we've got these fuzzy structures there. Those are the flower buds. And those flower buds open up to these beautiful yellow flowers here. They've got these yellow petals, and then you can see the stamens and the pistil in the center of that flower. And then here is the same plant, celandine poppy, a little bit later in the season. And you can see that the plant's a little bit leggier. Those leaves are fully opened. They are mature. But again, we've got these fuzzy structures here. They look, at first glance, they look a lot like, though they could be flower buds. But are they? If we look a little bit closer at that, we can see that they are different looking. So let's switch to the other one. These are closed up, fuzzy, small together. These are have longer stems that are more opened up. So this is actually not the flower bud. This is that developing fruit. And you can see that structure sticking out, and at the top of it, it's at the top of this picture, but at the base of that developing fruit, you can see kind of a brown or black circle around the edge where it hits the stem. That's where all of those flower petals had been attached and where they've fallen off. So you can see that. What we're seeing here is that swollen ovary, which again, in the other picture, that was in the very center. And then that part sticking out at the end, that's a remnant of the flower itself. So this is the fruit. It has similar features to the flower, but it is not the flower. And as is no surprise here, this is not anything earth-shattering, but that fruit is going to develop where that flower was on the plant. So they will grow in the same areas because it's that ovary in the flower that once it's pollinated, develops into the fruit and sets seed. So you'll see them in the same area. But if you know how to recognize the flower buds and the flowers and the fruits, you're not going to be confused when you see a structure like this. You'll know, okay, this isn't the flower bud, this is a fruit. So taking a look at bur oak, similar way of walking through it here. Bur oak is a wind pollinated plant like many other trees. Its flowers are not very showy. It happens to have male flowers and female flowers on the same plant. The male flowers release their pollen and fertilize the female flowers, and the female flowers are the ones that have the ovaries that are going to then develop into the fruit. In this case with bur oak, they're going to develop into acorns. So on the left here, we have a picture of the flower buds. These are male flower buds, and they come in these strings of flowers. And you can also see some of the leaves that are starting to break out of their leaf buds there. In the middle, this is a close-up of the end of one of the branches, and you can see at the top there what look like little baby acorns. Those are actually where the flowers are. And at the end, the parts that are dark brown, this is just beyond the flowers. So these are withered flowers that we're seeing there. That brown center part, that's where the flower was. And these are on their way to developing into the acorns. So male flower buds, female flowers that are withered. And then in the right hand, we've got the withered, dried male flowers there. And then again, we're starting with those female flowers, those female flower structures. They are going to develop into these acorns in the middle. And then, so this is early summer, and then at the end, we've got late summer. This is what those acorns look like. And you can see, comparing that first picture to the last picture, how they are visually similar, how that flower, that female flower, grows and expands into this acorn. And so if you can understand, if you can identify the differences between each of those different structures, the leaves, the flowers, and the fruits, you are going to be able to recognize whatever it is that's happening on your tree. A couple of things that it's good to get clear in your mind for whatever species you're looking at. Be able to tell the difference between the dormant bud versus the leaf bud or the flower bud. That can be helpful to understanding what you're seeing. Knowing the flower bud versus the open flower, what those look like, the developing fruit versus the ripe fruit, or the flower bud versus the developing fruit. So if you can identify these different phenophases, these different stages, you've pretty much got it covered. And those are the things that the questions are designed to help you make those observations and answer those questions. All right, just a last few things before I take some questions. I mentioned phenophase photo guides. On the right hand, this is one of our phenophase photo guides. This one's for tulip tree. It has pictures of each of those phenophase stages. We have, I think we've got about 45 to 50 of these that we've made for different species, and you can find these on our website. If you go on the menu at the top to the Learn tab and then select Phenophase Guides, it'll take you to this page over here, and you can click on any of those species, and that'll take you to where you can download the Phenophase Guide so that you've got that, and you can use that to help you figure out what you're seeing and understand what you're seeing on a particular plant. Something else that can help you become familiar with their species is using iNaturalist. So if you go on iNaturalist, you can search for a species and it'll give you photos of that species. That's what I'm showing here. And so this is Celandine poppy. It's a whole bunch of pictures that these are the observations people have submitted. So these are actual pictures of the species. I like to use iNaturalist and filter it. So in this second page right here, I filtered those observations. So it is just showing me pictures of Celandine poppy observations for Indiana. And it is just showing me observations for March. So this can tell me if I want to go out and find a Celandine poppy in March in Indiana, this is what I'm looking for. This is what I'm expecting to see. And so if you can use this to help you know what you should expect or what other people are seeing or what you would typically see at a particular point in time for your species. Or you can go to just looking at all the pictures and scroll down to see if you can to help you familiarize yourself with those different structures. So you can use this, look at a whole bunch of pictures of flowers. You can see what those flowers look like when they're fresh and new, or what they look like when they're starting to wither. You can see how that developing fruit begins to develop. And so iNaturalist is a really great tool for helping you become familiar with a particular species. Something else that can help you this year, we are doing some observer Q&A sessions. We've got one in May, June, and then we're planning on one in August and September. This is just a 30-minute come with your questions, bring your pictures that you are not sure what you're seeing, and we'll talk through it. So bring your questions, leave with your answers type thing, really informal. So if you are interested in those or you have questions, you can get to this, you can register for these sessions from our events page on our website. And then something else that you might find useful, one final resource, this you can search for on the Nature's Notebook website. They have a phenophase primer that they put together, and then also a botany primer that talks about all of those different structures in the plants, and then all of those different phenophases. These can be really helpful if you're interested in digging more into that. All right, I'm going to end there. I think I covered all of the things that I want to cover. I have a set of pictures here with the data sheet. We could practice if we wanted, or I can open it up and you can ask any questions, get whatever questions you have answered. So, any questions?
I have a couple of questions,
Sure.
if I may begin. My first question is, a lot of my trees are, like trees are, I mean, they're pretty tall. So I tend to observe the lower branches that I can see better. I'm wondering if I should use binoculars more to check the phenophases at the higher level of the tree, or is it okay to use the lower levels that I can see with the naked eye?
So there are two answers to that question. One, it is okay to focus mostly on what's lower, what's easier to see, to really understand what's happening. But as the questions are designed, it's one or more. So when you're at the beginning of the season, when there's nothing happening down on your low branches, you do want to make sure you're looking up to see if things are happening up there. So binocular is super helpful in that case. It's most important to make sure you look everywhere when you're in that range where things are either beginning to happen or about to stop, so that you get the actual answer. So if it's happening on your low branches, then great, you're good to go to just put that yes down. And you
Okay.
don't need to try really hard to look at every single last branch. But if nothing's happening down low, you do need to branch out and check and make sure things aren't happening somewhere else on your plant. Does that make sense?
Okay, yes,
Okay,
Okay, can I ask a second question now?
absolutely.
Is it good to have more than one specimen for each species? Like some of my plants, I've already noticed their phenotypes are a little bit different, like 2 green ash trees have a different timetable. So I've included 2 green ash tree specimens. But in general, would you prefer to have just one specimen or more than one of the same?
You are always going to get me saying more data is better than less, but some data is better than no data. So if you are noticing differences between 2 different specimens and you're interested in that, I would say go ahead and do more than one of a species.
Okay.
But if it comes down to if you either you add more and you observe more, or you're thinking, nope, that's too much, don't bite off more than you can chew. If you only have time to observe one specimen, do that rather than adding a second one and not observing as often.
Okay. Thank you.
Other questions?
Amanda, I I have a couple of questions.
Yeah.
One thing that drove me absolutely crazy, which is not hard to do, was the colored leaves. And one or more leaves show their color yellow or brown, or there are even spots, but not if the spots are due to stresses, drought, or disease. Well, how the heck am I supposed to know? I mean, I could go to almost the tulip tree almost any time of the year and find a spot somewhere on some leaf. And it seemed insane to say, well, here's a spot on one leaf. I'm going to report it as colored. So how do I know what's causing spots and when I should say, oh, this is drought or this is stress or this is disease and this isn't?
So the definition is one or more leaves show some of their typical late season color or yellow or brown due to drought or other stresses. So if it's late season color, yes. If it's yellow or brown due to drought or stress, yes. This is largely most of the leaf. The things you say you don't count it for are small spots of color, minor leaf damage, or dieback on branches that are broken. So if it's like an insect nibbled at it and it's just like a little circle around here and it's got some color around the edge, that's a no, you can ignore that. But if it's a full leaf, or all around the leaf, the whole leaf is showing some of the color, then that's when you're going to say yes. And it is helpful to report one leaf that is drought or stress related coloration that you're seeing, because that tells us if that plant is stressed, it may have a different timing for its phenophases than a plant that is not stressed. So some of those at Holiday Park, some of those plants, almost always, I'm saying yes to colored leaves, particularly the service berries. There's always some colored leaves, and it's okay, say yes to those. But if it's just a little bit of insect damage or just a corner of the leaf, that's when we're saying no. Does that answer the question?
Yes, well, I think so.
Good. Other questions?
Could you go back to, and maybe I can figure this out just by going and looking at the website, but you said something was a leaf instead of a leaf bud, and the last phase, it looked pretty much still like a bud to me. The leaf was all folded up and I couldn't see any stem. So I was a little confused by that. Exactly when does a leaf bud cease to be a leaf bud?
Okay, was that on the breaking leaf buds
Yeah,
slide? Okay. This one right here.
yeah, that last one.
Okay, so that last one, you can see the stem in there. Let's see if I can get this. So right in here, you can see the stem on that part. And that's when you can see that stem attachment right there. That's when you say a no. So this one is a no to breaking leaf buds.
Okay, so when the green attaches to the twig, I guess.
Yeah, so it's when it's unfolded. So if we go to this, gotta click on the right spot. This next one here, so the leaves, when it's in its breaking leaf bud stage, it's not a yes to leaves. It becomes a yes to leaves once it's fully broken out of its bud. And so here a leaf is considered unfolded once its entire length has emerged from that breaking bud, stem, node, or growing stem tip, so that the base of the leaf or the leaf stalk is visible at its point of attachment to the stem. So when it becomes unfolded, that's when you start saying, or you stop saying yes to the breaking leaf buds.
Okay, and then one final question, just out of curiosity. I assume all this data goes into a huge computer data bank somewhere. And so I'm figuring, why am I making comments? Nobody is going to stop and read a comment when there are thousands and thousands of data points going into a computer, and nobody's going to care whether one red bud has a lot of leaves still and another red bud I'm observing has lost most of its leaves. Who's going to read that?
So I read it. It all goes into the national database. There are more than 10 million data points in that database. So you're right. Some researcher from clear across the country who happens to just be looking at oaks knows you're looking at some oaks. They pull your observations with hundreds, thousands of other people's observations, and they probably aren't going to be looking at your comments. But when I'm looking at everything that happens at Holiday Park, I do look at those comments, and I use those when I am writing reports about what's happening at that particular location. So comments are good. I like them. I'll read them, but by and large, anybody else that happens to be looking at it from someplace else, they're not going to read those comments.
Okay.
We are now after 1 o'clock. Any last questions before we say goodbye and close it down? All right. Thank you for joining me. Thank you for making observations. Again, if you have any questions at any time, email me. I am happy to help. Want to make sure you are feeling comfortable in making these observations, and I really appreciate it.